So now I would like to continue with the program. And so our next speaker is Roger Thilmony from the USDA. And he's going to talk to us about his time in the 2000s here at the PRL. Thank you, Roger.
Thank you. So first, my congratulations to the PRL on 60 great years. I want to thank Christoph Benning for the invitation and the committee that invited me. And I had a host, Bailey, hosting me, and we went out to dinner last night with Greg Howe. That was great. It was great to catch up with him. And I'm going to give you my perspective. So I'm supposed to represent the 2000s, but I'm going to start here. And because this is a famous image that actually I remember Hans Kendi showing to us, I think it was in a noon seminar at some point, and talking about the original faculty of the PRL. When I joined the PRL, that was in 1999, January of 99, and there were still three of the original faculty working as part of the team. So Peter Wolk, Jan Zevaert, and of course Hans Kendi. And it impressed me that all three of those guys were still hard at work in the lab almost daily when I was here. So, but how did I get to the PRL? Actually, I graduated from Purdue University. I was in Greg Martin's lab at Purdue, and he's, of course, an alumnus of the PRL, too, as a graduate student. And so when my wife and I, who met across the bench in graduate school, got married, we were a two-body problem again, looking for two postdocs in the same place. And there aren't that many places in the United States or even the world that have two postdoc positions open at the same time in the topic areas in which we were working. But one of them, so when we investigated the PRL, there were two relatively new faculty. Look at these old pictures, right? Sheng Yang He had recently joined the PRL a few years before that, and I think Greg Howe was maybe in his second year at the PRL. He could correct me if I'm wrong. And interestingly, their topics matched both my wife and I pretty well. So we were in Greg Martin's lab. We were working on plant microbe interactions, particularly Pseudomonas syringae's interaction with tomato. And so we had interest of continuing work in plant microbe interactions and or working with tomato. And of course, these two labs pretty much fit that bill. And so I interviewed with Sheng Yang, and she interviewed with Greg. And she actually graduated about 6 months or 9 months before I did. We walked at the same time. So this is a picture of her graduation in December of 98. And she had already been living up here at that point, living in East Lansing for about 6 months and working in Greg's lab. And then I made the trip and moved to East Lansing as well. It's about a 4-hour drive. I can tell you I made that drive quite a few times in those 6 months because I was pretty lonely down. Because at that point, Greg Martin had recently moved his lab from West Lafayette in Purdue to Ithaca, New York at Cornell. And so it was me and one other grad student that was left behind in his lab working there those last few months. And of course, I found an old version of a presentation I gave, I think at a noon seminar. And so these are probably out-of-date logos for the PRL in Michigan State. And so I'm going to talk a little bit about the perspective of how I ended up in Sheng Yang's lab here at the PRL. And Sheng Yang at that time was mostly known for his work on the bacterial side of the plant microbe interaction, of working in Pseudomonas syringae and studying the genes that are responsible for producing the type 3 secretion system and the effectors and conferring disease in plants. But his lab was actually making a transition at that time to studying a different aspect of that interaction. And basically, you heard from Scott a lot about innate immunity, the resistance plants have to bacterial and other pathogens. But what was new at that time in Sheng Yang's lab is he was pursuing the idea of trying to understand better how do these pathogens actually cause disease in plants? So how do they colonize the plant to be a successful pathogen and multiply and cause disease? And so very little was known in the early 2000s on that. And there were two main hypotheses. And interestingly, both of these hypotheses turned out to be substantiated later by research both in Sheng Yang's lab as well as many other labs. And it basically showed that effectors that come from the bacteria have two main functions. They are suppressors of plant defense response, or evaders in some cases, and or they promote nutrient releases, so releasing sugar, nutrients, and other nutrients that promote the bacterial ability to grow in the plant host. And when I joined the PRL in early 99, January of 1999, what was super hot back then, this really dates it, right? If you see that picture, that's a cDNA microarray. So its DNA is spotted onto a glass slide, and this was global transcription profiling, which was the new and hot thing at that point. There was the Arabidopsis Functional Genomics Consortium being led in the PRL, and they had all these ESTs, this huge collection of expressed transcripts from Arabidopsis that they could spot onto the slide, and you can look at the expression of tens of thousands of genes at a time. And of course, I wanted to apply that technology to studying plant pathogen interactions. And so I'm just going to go through and tell you a little bit about how I remember the plant research lab. And one of the first things I remember is a very dynamic seminar schedule. So this one I just pulled from the annual report, 1999. I don't know if you can read all that, but there's some eminent speakers that they would invite every year to the seminar series. And so there was a very interesting seminar series that we held every year. But the one that makes the biggest impact, at least on me as a postdoc or probably grad students that are in the PRL, is other things like Choices Day. So I don't know if you guys still do this in the PRL, but Choices Day was when we got an opportunity to think about our career paths. So of course, the obvious path, if you get a PhD in science, is you become a faculty somewhere at a university and teach classes and run a research lab. But it was pretty obvious to us at that time in the early 2000s is that there weren't enough faculty positions for us all to have that position. And so this was one thing the PRL did that really exposed the students and the postdocs to the options of what other career paths are there for people with our training in science. And not only did I benefit by participating in the Choices Day that was held when I was here in 2000, but I actually was invited back, I think in 2009, maybe, to be a representative of government employee. So I work for the United States Department of Agriculture. Actually, I think my job just got reactivated today. I was on furlough for the last month and a half, or what I consider forced vacation. So, and I'm here on vacation right now. I'm not here for work. But basically exposing you to these options, whether it's working for the government, working for a research institute, working for lots of different options that we could pursue as our career. And the other thing that I really remember when I think back on my time at the PRL are the noon seminars, right? I think you guys still do the noon seminars, right? So, and my knees wobbling when I'm standing up in front of, I've very much noticed that going and giving a talk at a conference where you know almost no one in the audience is way less stressful than giving a talk in front of everybody that knows you and sees you every day, right? So including your own lab members as well as the neighboring lab members and Hans Kendi or Jan Zavart raising their hand and asking you a question in a noon seminar about hormone signaling, and you're going, maybe, let's talk about that afterwards. So, but this was the one I found from the annual report where my name's on the bottom of the right. That was my first noon seminar, and it talks about the microarray analysis of compatible interactions between Pseudomonas and Arabidopsis. And so I'm just going to talk a little bit about that science. I realize this is 23 years old now, but it's the interaction between what we've heard about from Scott, between these two organisms. And the reason I chose this image is if anybody works in Arabidopsis and Pseudomonas interactions, you've probably seen that picture a lot of times. And I took it. so it's really hard. I don't know if you've worked in plant pathogen interactions. Trying to get good pictures of diseased leaves is amazingly hard to do, right? Because you got to get the color balance right. And in Arabidopsis, leaves tend to die very quickly when they're susceptible to the pathogen. But if you get it just at the right time, these are the symptoms you see with Pseudomonas and Arabidopsis, which is first chlorosis, the yellowing of the leaves, the loss of chlorophyll, and then the necrotic spots that you can see on those plants. And then it made it onto the cover in 2001 onto the annual report for the PRL. And I've seen it in multiple reviews from Sheng Yang's group as well as a few others, actually. So, and so this was the work I worked on, is basically trying to understand using global transcriptomics of these old-style cDNA microarrays to identify genes that are induced and repressed in response. And the colors here, red is induced, green is repressed, and yellow is basically constitutively expressed between the treatment and the control. And interestingly, you don't realize how much of a problem it is. It used to be, when I started my postdoc, finding one responsive gene, you thought you hit the jackpot, right? With this technology, we soon had hundreds of genes, which instantly turns to, instead of hitting the jackpot, to being overwhelming, trying to figure out what it all means, right? So, but one of the interesting things that we discovered from this process is not only are the induced genes important and interesting in the phenomenon of the interaction, but actually the repressed genes gave us a hint of what the virulent bacterial pathogen is doing, and it's suppressing the defense responses, and particularly those defense responses of genes that encode proteins that are related to secretion. So secretion of defense responses from the plant cell, trying to stop the ability of the pathogen to colonize. And so I want to make a shout out. I've been very lucky in my career. I've had great mentors, and Sheng Yang He is definitely one of those great mentors that I've benefited from. He is a very generous scientist, very smart, super helpful and supportive, and I very much enjoyed my time working in Sheng Yang's group. And these are the people that overlapped with me while I was in his lab from '99 to 2003. Shruti, Elena, Paula were all graduate students. Chaoling was another postdoc that overlapped pretty much exactly with me. Ola, Youngbom, Youngnam, Kenya. You guys probably know Kenya. He was here for a really long time. He came when I was a postdoc, and I think he stayed with Sheng Yang when he moved to Duke. Anne was the lab manager. There's me. There's another grad student, Bill, and Julie, and many, many undergrads. And I realized at the time when I was trying to prepare this talk, I was trying to find great pictures of the lab, of us working in the lab or something, and I had to struggle to find them. So these are the best ones I could find. But these are mostly lunches that we took to celebrate something. And I didn't go through and name everybody, but most of the people you see in the pictures are members of the He Lab, or maybe honorary members, like that 2003 shows John Scott Craig. You joined us for lunch that day. But if you can't tell who's who, I don't know if I can get this to work, but that's me without the beard on the left in the 2003 picture. So I don't know if it changed too much, but that's me there. And we also attended multiple conferences as a lab group. One was in Madison. I think this was MPMI in 2001. That's Shruti and Chaoling and Julie and me, and I think Chaoling's fiance at that point. And then this is just a picture. You remember when we used to go take posters to meetings when they're in panels? That's what we did in 2000 and 2001. That's Chaoling and Kenya by their poster at that meeting. And so the reason it took me a long time to find these few pictures is that, oh, I'm getting ahead of myself. So one of the other things I think of when I think of the PRL is the collaborative and dynamic interactions we had between groups, not only within Sheng Yang's group, but also between other groups at the PRL, and how everybody was supportive and interested in potential collaborations and working together. One I already mentioned, the Arabidopsis Functional Genomics Consortium with the cDNA microarrays allowed us to make our own subarray with just the genes that were regulated that we were interested in. But another one I'm going to point out is a collaboration that started while I was a postdoc in Sheng Yang's lab, and that was with Greg Howe's group, and particularly members of his group including Youfu Zhao, Lei Li, and Chuanyu Li, and multiple others. And it was this connection that we made at that time between what Greg's lab was working on, what Sheng Yang's lab was working on, and this idea that there's a crosstalk between the signaling pathways when we recognize Pseudomonas. And not only does it secrete effectors, Pseudomonas syringae also produces a host toxin called carnitine, which is a jasmonic acid signaling mimic, and it activates the JA responses. And we work together between both labs to show that these two pathways are interacting during this response to pathogen attack, and it actually leads to the suppression of defense genes and the induction of wound response genes, which is another way that the pathogen suppresses the ability of the plant to fight off its infection. And that led to actually one of the quickest manuscripts I had in my career, probably. We started the work, and I think we finished it in less than a year and a half, which is pretty fast for a plant-based research project. The other reason that I had struggled to find lab pictures is I was here in 1999, 2003. My wife and I got married in 97, and when we moved here, we had our first child in 1999. And that's a picture of Ying-Su, my wife, and our first son, Alec, in front of the Bailey statue. Is it still there in the garden? Yeah. So that's, of course, most of what I was taking pictures at that time was of Alec and her. And this is just a few more cute pictures I found of. And then in 2003, we had our second child, Luke. And he's held there by Alec, and they're together there. And he was born in April. And then in June, I accepted a position with the USDA, and we moved from East Lansing to California. And so that was an epic drive with a baby that was 3 months old. So I think there was something in the water in the Sheng Yang He lab, because I wasn't the only one. My wife and I weren't the only ones that had babies while in Sheng Yang's lab. That's Chao Ling with her daughter, and Kenya with his daughter and family. And then after I left the lab, there on the right, Elena, Julie, and Nele each had babies as well. So it wasn't just me that was adding to our family at that point. It seemed to be a trend in the He lab. Okay, so moving from the plant research lab, I accepted a position in 2003, and we moved to the Agricultural Research Service. It's in the San Francisco Bay Area, so that's a picture of our family in front of the Golden Gate Bridge. And we quickly bought a house, and I've actually been there ever since. But this brings you to the present day, almost. So this is a picture of our two sons. They're on the right there. So Alec, he graduated from college 4 years ago, and he's working for a biopharmaceutical company in the Bay Area. And Luke graduated with a degree in statistics in June and is still looking for a job. You need a statistician? Let me know. I'll connect you up with him. So, and then my wife and I are there. So that's the transition that's happened. Okay, that's what I have for the PRL. I'm now going to switch gears. So my interest in plant science actually comes from my background. I grew up on a farm in North Dakota, so I have a genuine interest in agriculture and applying science to make our agricultural better. And that's why the job at the USDA interested me at the time when I accepted it. And it's because we've been focused on, for now 20 years, developing molecular tools to allow us to better engineer crops to make them better. And so today, we have multiple projects in the USDA and the project that I'm lead scientist of. One of them is to characterize a suite of useful crop promoters. So promoters that can express your gene interest precisely in the tissues or under the environmental conditions you need to do your engineering. But I'm not going to talk about that work today. I'm going to talk about a different effort that we made over the last 6 or 7 years, developing a system we call gantry, which I hope I'll convince you is an effective and efficient way of doing transgene stacking in crop plants. And so we've been genetically engineering plants since the late 80s, but putting 1 or 2 genes into plants using agrobacterium is a pretty simple thing to do. You can build a construct in a day or 2, put it in agrobacterium, and undergo tissue culture transformation and to generate it. But if you're trying to engineer pathways or complex traits that require suites of genes to do this, then the technologies become more challenging. And so what I'm going to show you today is how I hope the gantry system can enable everyone to be able to engineer multi-gene traits into crop plants efficiently. And so why this name gantry? A postdoc in my lab, Ray Collier, came up with it. I have no credit for this, but I'll tell you what it stands for. It stands for Gene Assembly in Agrobacterium by Nucleic Acid Transfer using recombinase technology. I actually have to look at it to make sure I say it right. So I don't expect you to remember that. But it is a pretty nice analogy, and that's what's shown in the picture here. This is a gantry crane in red and white. And so what those cranes can do is they can load these cargo containers onto a container ship. And so you can think of the gantry system as a way of allowing us to load lots of cargo into a transformation tDNA construct. And so instead of being a ship, the ship is agrobacterium, and the system of the crane is actually these recombinases that allow us to efficiently stack these traits together. And so I think of gantry as a sophisticated system, but it's actually simple in the sense it requires only 3 types of components that is easy for the user to implement. The first is you start with a modified agrobacterium strain. The data I'm going to show you today is a strain we call rport1, which is the original strain we engineered to be a gantry recipient. And what it basically does is you modify the virulence plasmid, so removing the native tDNA, and replacing it with a system that has a bacterial selection marker flanked by these recombination recognition sites. And I'll talk more about those in a minute. And that enables you then to stack into the native virulence plasmid. So essentially what you're doing is you're removing the native tDNA and replacing it with the tDNA with the cargo that you intend to introduce into plants. And the other 2 components you need, the first are 2 donor plasmids. These are just cloning vectors. When we originally developed the system, we just made them, they're basically puck-based plasmid backbones with a large multiple cloning site. That was restriction enzyme-based cloning. Almost nobody does that anymore. So we've now modified and are typically using Golden Gate cloning to do the assembly into the donor plasmids, or you could use other technologies like Gateway or other technologies for building your plasmids. But the idea is you just take your cargo sequences of interest and introduce them into one or the other plasmids, either the B or P donors. And I'll explain why we have 2 different type of donors there. And we also have 2 types of what we call helper vectors. These plasmids are not modified by the user, they're just used. And we call them helpers because they provide the site-specific recombinase enzymes that actually physically do the stacking into the virulence plasmid. And so B and P are the 2 types. And so the idea is we can do stacking now in vivo. So this isn't in a test tube in the sense of just DNA into a plasmid. This is actually in vivo into a live agrobacterium strain. And the method enables us to sequentially stack. So you can't just do, you can do multiple rounds of stacking. So you can sequentially stack as much cargo as you need to into the tDNA construct. And it's based on the use of these 3 powerful enzymes, which are unidirectional site-specific recombinases. So PAR-A, TP901, and A108. So what PAR-A does, it recognizes 2 sites called multi-resolution sites. These green arrows called MRS there. And when PAR-A sees those 2 sites, what it does is it does a recombination reaction, excising the intervening sequence between the MRS sites, leaving a single MRS site behind in the substrate that you gave it. And that excision reaction is important. I'll tell you how it works together with the other 2 later. The other 2 recombinases, TP901 and A108, are different. PAR-A does an excision reaction. These other 2 recombinases do a unidirectional integration. So you give it a target and a substrate that has 2 recognition sites called at B and P. And it performs integration reactions where you integrate those 2 sequences together. And then it produces modified sites that are no longer recognized by the recombinase. So they're stable integrations. They can only go that direction. And so when you take these components and put them all together, this is basically how the gantry assembly process works. You start with an rport strain that has the original target sequences integrated into the virulence plasmid. You then electroporate that strain with a B donor carrying whatever cargo of interest you want. It could be 1 expression cassette, it could be 2, 3, or 4 expression cassettes, however many you want, with in the B donor. The B donor recognizes its at B and P sites, and it's co-transformed with the helper vector that expresses that recombinase. And what it does is it performs an integration reaction, followed immediately by an excision reaction, which essentially removes the original components and now has replaced it with a tDNA. LB and RB are the border sequences on the tDNA that have your cargo sequence, and it leaves an at B site for the other recombinase, TP901, behind. And what happened is you started with a kanamycin-resistant strain. It's now gentamicin-resistant. You now then can repeat that process exactly the same, but using the other donor, a P donor and a P helper. Integration and excision will occur again to produce your stack 2 strains. So it's now a tDNA that has both of those cargoes, and you've now converted it back from gent back to kanamycin resistance. So if you're paying attention, you can repeat this process as many times as you want to build as big a tDNA as you'd like. And so that was the design in theory. Now I'm going to show you in practice what it worked like. So when we first developed this system, we wanted to demonstrate how well it could do this stacking in vivo in the Agrobacterium strain. And so we decided to perform 10 sequential sequence stacking events and built a 29 kilobase tDNA. I'm not going to walk you through all the cargoes that we inserted. We basically just picked cargo components that has an easy assayable phenotype in a transgenic plant. And if you add those all together, and we did 10 rounds of stacking. So stack 1 had the number 1, the 35S promoter on a sulfadiazine resistance gene and the 35S terminator. And then we kept toggling back and forth between B and P donors that have additional cargo to build. This is what what we call the 10 stack looked like when we got completed. And so what I can tell you is that each round of stacking, and when you undergo the selection from kanamycin to gent and back to can, essentially the stacking is 100%. So you don't really even have to check the colonies to have their cargo. If they have the antibiotic resistance and the loss of a counter marker called SAC-B, a sucrose sensitivity marker in agro, it essentially has your cargo. So when we first did this, we checked everything with PCR at every step. But we don't even do that anymore because it works 100% every time. And so this is what the first 10 stack looks like. And of course, I mostly work in crop plants. We do genetic engineering of crops primarily, but Arabidopsis is so easy to do transformation. So we first tested this in Arabidopsis. And so we just did floral dip transformation with the 10 stack and recovered plants at about 1% efficiency that were co-resistant to the 2 selection markers that are on each edge of the tDNA, the sulfadiazine marker on the left border side and the kanamycin marker on the right border side. And so transformation efficiency was surprising. I was expecting maybe the transformation efficiency would be lower because of the size of the tDNA, but it was actually only a modest bit lower, maybe 1%, 2% to 1% is what's normal when we do Arabidopsis transformation. And so these are the phenotypes we saw in these transgenic plants. So resistance to antibiotics, green fluorescence in the pollen, gust staining in basically all the tissues. We had a MIB transcription factor that activated anthocyanin expression in the petals and the seeds, and red fluorescence is shown there on the right panel. And so the postdoc working on this in my lab was very ambitious. He characterized over 434 transgenic events, and 90% of the events had all 8 positive phenotypes. So the fidelity of getting the whole tDNA construct into your transgenic plant was quite high. And 93% of those lines carried only the tDNA and no sequence outside the border regions of the tDNA. So the quality of the events, that is, it specifically got the sequences we wanted it to introduce and not outside of that, was also very high. And we then used droplet digital PCR to measure transgene copy number for 3 of the components in the 10 stack. The first one is the SUL1 marker on the left border. And what you can see is almost 70% of the plants we recovered had a single copy of the SUL1 sequence. If we analyze the copy number for GUS and MPT2, you can see a trend there. And what that basically shows is that we can easily get complete tDNAs, but some of those events actually also contain partial tDNAs that initiated at the right border, but failed to get all the way to the left border and get the complete tDNA integrated. And so, but even among those lines, 27% of the events were a single copy for all 3 across that 10-stack tDNA. So those events essentially had 1 complete tDNA integrated into the genome. Okay, Arabidopsis is a great model. I'll be the first to say that. But my lab primarily works in crop plants. So the next question we wanted to ask is, how does this work in crops? And so a colleague's lab took the 10 stack and could directly transform that into potato, and they tested it. I'm not going to show you the data today, but it works very well in potato. I'm going to show you what we did in rice. The 10 stack we built for Arabidopsis mostly uses promoters from dicots or promoters that work well in dicot plants. But a lot of those cassettes, expression cassettes, won't work very well in rice. So instead of using the 10 stack, we went and built an entirely new stacked tDNA that now has 11 stacked components. Some of them have the same genes, but just different expression elements to control their expression. And they're all listed here. But this tDNA, even though it's only an 11 stack, it's actually quite a bit bigger than the 29 kb 10 stack that I showed you before. It's almost 37,500 base pairs. And we did the same thing. It was just now in rice, and analyzed the phenotypes of these plants. And this just shows you some of the phenotypes we see: luciferase activity, both Ranella and firefly, herbicide tolerance, antibiotic resistance, green fluorescence, gust staining, and did the same characterization, but this time on a more modest number of plants. We didn't do 400 rice plants. We did about 40 transgenic rice plants. And this diagram just shows you the fidelity of full, complete tDNA integration in these 40 plants. And so when you see the bar underneath the diagram of the 11-stack tDNA, if that's there, that means based on PCR or functional data, we know that that sequence was present in the transgenic plant. And what you can see is that basically the top half of these 37 events, none of these events had backbone sequence. So again, it just had high fidelity for integrating just the tDNA and not unintended sequences. But about half of these plants had all 8 of the assayable phenotypes. And in this case, we only used a single selection agent to identify these plants, and that's the hygromycin resistance gene near the left border. And 22% of these plants were high quality because they were single copy for multiple components of this 11-stack tDNA, and they had all the phenotypes. And so we published this work a few years ago, but since then we've gone on and take 2 of these events, number 9 and number 42, and it's gotten so easy to do whole genome sequencing, long read sequencing. We took 9 and 42 and did PacBio read sequencing on these 2 transgenic events to thoroughly characterize these 2 events and to see what they really look like in those two plants. The first one with the 11 stack, actually, it's nearly perfect. You have 37 kb of the tDNA. It's only missing 261 base pairs near the left border, but that selection marker is functional because we used it for selection. And it was inserted in the intragenic region, so between genes, on chromosome 6, and it only caused a very small deletion, 64 base pairs of the native genomic sequence. A similar story for the 11-stack number 42 event. It's also, again, almost complete, just missing 285 base pairs near the left border, and it integrated into chromosome 9 with a very small deletion of the native sequence, 17 base pairs. So what I'm trying to convince you is that this technology can produce high-quality transgenic events, not only in Arabidopsis, but rice, potato, and numerous species if you need to engineer multiple traits into your plant. And so I won't belabor this point, but this is just a summary of the results we got in rice. It just shows you the frequency of the positive phenotypes that we observed in the 37 plants and summarizes that for you. And so Arabidopsis, potato, rice, I've already talked about those. But we've gone on and now done a model grass, Brachypodium distachyon. We've used the 11-stack in switchgrass. A colleague of mine has transformed citrus using large stacks of constructs, both into oranges and limes. We've done tomato. We've done grapevine most recently in my lab. And then through collaborations or just people that have received the gantry technology from us, have done soybean, wheat, corn, poplar, pennycress, and probably other species I haven't been told about by the users. And so I hope you're convinced that this technology is an efficient way of making stacked plants with large numbers of genes or traits, and it can work well in a wide array of species, and it frequently will develop high-quality single-copy events with no unintended sequences. And I showed you today that we could build constructs up to 37 kb, but what I didn't have time to, but we've now built tDNA constructs over 100 kilobases in size. It's not a limit of what you can build. It's a limit of what Agrobacterium can efficiently deliver into the plant. So we can build really large tDNA constructs, but then getting that whole 100 kb tDNA into a plant in an efficient way is the bottleneck right now. So definitely anything under 50 kb is not really a problem. Anything over about 75 or 80 kb, it gets pretty hard to recover a plant with all that sequence. Okay, and as I alluded to already, I hope this system is not only useful in my lab, but I hope it could be useful in many other groups and many other research labs. I work for the government. It's publicly funded research. We create these tools to be used. So please feel free to contact me. This is just an example of some of the organizations we've delivered the gantry system to. If you'll notice on the upper left there, it's Michigan State. So I had to include this part. Unfortunately, it was not someone in the PRL, but it actually was David Dauch's group. And it's a large collaborative group on Feed the Future. And they use the gantry system in potato to build a pretty large stack. It's shown here. They have a kanamycin selection marker in the tDNA, but other than that, all the other components are actually disease resistance genes. So they had 3 different late blight resistance genes, so resistance to Phytophthora infestans, from wild species of potato, essentially, as well as 2 virus resistance genes. So they built a almost 35 kb tDNA, and actually, just a year ago now, they tested these in the field and showed that they're functional. They confer resistance to the target diseases. And then I think it's a year ago, June, it was actually, they received regulatory status review approval by USDA APHIS so that they could grow these in the field. So this is just an example. I love to see this. My brother is still the farmer. He still lives in North Dakota on the farm I grew up on. He doesn't grow potatoes, so he can't grow this, but I'm hoping someday this technology will get deployed in a crop that's grown here in the US. Okay, so I just want to acknowledge all the hard work. The 3 highlighted there. So originally, Ray Collier was the postdoc in my lab who first was part of the idea for the gantry system, and that also involved James Thompson, a colleague in my USDA ARS project. And then the rice work was done by Leila Haithwaite, and a lot of the PacBio sequencing and everything was done by my lab manager, Tony Moore, as well as the other collaborators who were involved in different parts of this project. But I would be happy to answer any questions.
Thank you, Roger. Great talk. Any questions for Roger?
What is the long-term stability of these constructs?
The Arabidopsis and rice plants we've taken to the T4. There doesn't seem to be any problem for the single copy tDNA integration events. If you have multiple copies or partial tDNA integration, sometimes you'll get gene silencing. But as long as that's not occurring, the stability is good. Yeah, that's the high quality event thing. So if you get one complete copy as designed, that usually is quite stable as a trait. Yeah, good question.
Nice to hear about Dave Dauch's work, and it just makes it tragic that USAID canceled the funding. Is there a chance that there can still be outreach and potato strains offered to other countries?
I wish I knew the answer to that question. That's a great question to ask David. I hope so. The other issue that's impending on this, I said it got RSR review, but since then there's been court cases that have impacted the way regulatory system works here in the US again. So we're back to the old system. So it got review and approval based on the new dogma of how regulatory review was happening. But there was a court case a year and a half ago, I think, that put us back into the 1990s as far as the regulatory review went. So yeah, there's a lot of issues there. I love to see this technology in the field and helping researchers get their genes of interest, their traits of interest into crops in the field is the goal. And I hope we can get there. And I wish there were easy answers, unfortunately, not usually. So, but good question. Yeah.
Have you tried it in sorghum? Asking for a friend.
I'm working with Peggy Lameau's lab.
Okay.
We're working on doing it in sorghum right now. So Peggy's lab primarily uses a different agrobacterium strain, and they use basically the Corteva system with baby boom bushel.
Yes.
And so there's a lot of restrictions on how you could deploy anything like that. So we're essentially trying to create a system that would be gantry-based with bushel expression so that they could get efficient transformation in sorghum, and yet it wouldn't be impeded by the intellectual property that Corteva holds on their technology. Yeah. But yeah, we're working on it right now. They predominantly use an Agrobacterium tumefacient strain called LBA 4404, so we're essentially converting that strain into a gantry recipient so that she can stack their cargo into that strain and use it. Yeah.
Okay.
Yeah.
Let me know if you need help.
Ask me in a year, or Peggy in a year. I hope we'll have the answer to that.
That's promising.
Yeah,
Thank you, Roger. Any more questions? If not, please join me in thanking Roger again. Thank you.